Hello friends, welcome to CEC Live Lectures. Dear friends, taking our series forward on cell biology. Today we are going to talk on cell signaling, and for the discussion on the topic we have with us in our studios Dr. Varunendra Singh Rawat. Dr. Varunendra Singh Rawat is Assistant Professor in Department of Zoology and he is teaching in Hindu College, University of Delhi. Dr. Rawat has immense experience, so let's take advantages from his experience and let's try to understand this topic in detail, that is cell signaling. Hello sir, welcome to the lecture.
Thank you. So friends, we always see that people interact with each other, people communicate with each other, and for that they require certain signals to be given by them and for the perceiver to receive those signals by using sense organs. And the success of any species depends on successful communication between the members. But when we talk of the organism itself, when we do not talk at the population level, when we talk at the organismal level, how do various cells of the organism communicate with each other? Because the success of any organism, for the organism to live for a long duration of time, it depends on the successful communication between various cells which form various tissues and organs. So how do different cells at different location of the body of the multicellular organism communicate with each other? So I'll be talking about different methods or different ways by which different cells communicate with each other in today's lecture, cell signaling. So what exactly constitutes cell signaling and how do different cells perceive the signal being given by other cells? So today's lecture will cover the basics of cell signaling. What are the different signal molecules which can be used as signals by various cells, and how do various cells receive these signals with the help of what type of receptors they receive these signals, and what do they do afterward? That is the topic of discussion today. So after this lecture, we should be able to tell about various types of signaling like autocrine, paracrine and endocrine signaling, different types of signal molecules which are used in the multicellular organism body, and different types of receptors. So starting with the basics of cell signaling, we all know that the cell is surrounded by a large amount of extracellular material, and apart from the extracellular matrix which surround the cells, there are certain other molecules being secreted by other cells which can be perceived by individual cells, and a cascade of reactions follows after that. So the messages which are being given by specific cells, they can either travel to long distances or they can travel to short distances. Suppose a hormone is being produced in the pituitary of the brain of an individual and it has to affect the gonads. So how does that particular signal travels from the pituitary, which is located in the brain, to the gonads of the organism, which is located quite a far distance away? So what exactly happens here, and how exactly the cells are stimulated after receiving these particular signals which are emanated by different types of other cells? For these cells to accept these signals, they have to have certain kind of receptor mechanism, and once they receive this signal, they will ensure that the cascade of reaction which follows afterwards, it ensues. So we can divide, or we can categorize the extracellular messenger molecules which transmit messages between different cells into three types. The signaling can be divided into three types: autocrine signaling, paracrine signaling and endocrine signaling. As the name suggests, autocrine signaling is basically a signal is being given by a cell and it is received by that particular cell itself. The same cell receives that signal. That means the cell has a receptor for the signal being given by itself, and it is seen in a number of lymphocytes where they emanate the signal. The receptor on the cell itself bind that signal, and this helps in proliferation of these lymphocytes. Another mode is paracrine signaling. Now, what exactly is paracrine signaling? It is basically a release of the signal by a cell, and this particular signal is being deciphered or is being accepted by cells in the vicinity of this particular cell. That means the messenger molecule or the signal molecule does not travel to a very far off distance. It is just in the locality of the cell which is receiving that particular signal. And the third is endocrine signaling. Now, this particular mode of signaling is the one wherein the signal molecule travels to the greatest distance. And this particular thing or mode happens because the signal molecules travel through the bloodstream from the the site of origin of this particular signal to the site where this particular signal is received. So, this particular panel shows how these three modes are different. It is a simplistic way of representing the autocrine signaling, which is the A figure. The cells are receiving the signal themselves, and paracrine signaling where the cell is emanating the signal and it is being received by the cells which are nearby. And third is the endocrine signaling, where the signal is released into the bloodstream and it travels to far off distances where the receptor receives this particular signal. Now, what categorizes as a signal molecule? What exactly is a signal molecule? Which molecules can be used as signal molecule? So, there is no fixed category, but there are it's not that only carbohydrates can be signal molecules or steroids can be signal molecules. There are a vast array of molecules which can be utilized as signal molecule. These could be small amine hormones or small protein hormones or very big glycoproteins which could be present in the extracellular environment of the cell. The cells have to receive these signals. The basic criteria for a signal molecule to be a signal molecule is that it should have a receiving mechanism in the target cell. So, cells should have receptor which should receive these signal molecule, and then they lead to the further cascade of reaction. The signal molecule which binds to the receptor on the receiving cell is termed as ligand. Now, this if we say that two cells have similar type of receptor, do they have to have a similar type of response? That means when two different types of cells bind the same type of ligand with the same type of receptor, is it necessary that both these types of cells have to have a same set of processes afterward? It's not always the case. For example, if we take liver cells and smooth cells, and if we take beta-2 adrenergic receptor in these two cells. Now, these two receptors, that is beta-2 adrenergic receptor, it binds the adrenaline molecule. But the effect which adrenaline produces in these two types of cells is quite different. In liver cell, binding of adrenaline by the beta-2 adrenergic receptor leads to breakdown of glycogen in the liver. Whereas in smooth cells, whenever beta-2 adrenergic receptor bind adrenaline, it causes relaxation of smooth muscle cells. So, we have two types of cells, we have same type of receptor, we have same type of ligand, but the end result is different in both the cases because the cell types are different. So, each type of signal molecule can produce different effect in different types of cells depending on the downward cascade after the binding of ligand to the receptor. So, it depends on the type of cell and it depends on the environment or the requirement of that particular cell, that what exact end processes occur after binding of the ligand to the receptor. Now, these signal molecules, they have two ways of going into the cell or transmitting their information. There are some signal molecules which bind to the cell surface receptor, and this generates an intracellular second messenger through another enzyme known as effector. And the other mechanism is that the surface receptors recruit some other proteins to their cytoplasmic domain. In most of the cases, the receptor which is going to bind the ligand molecule or the signal molecule, it has its cytoplasmic tail. Near the cytoplasmic tails are present some other proteins which will get activated after the ligand binds to the receptor. Once these cytoplasmic proteins are activated, they will ensure that other proteins in the cascade are activated and a set of reactions take place. But other type of process through which signal molecules can transmit their information is by binding to the receptor, and instead of activating other proteins, the cytoplasmic tail attracts some other protein to the cytoplasmic tails, and afterwards the downward cascade is initiated. Now, most of the signal molecules, they lead to formation of certain molecules which are termed as second messengers. These are termed as second messengers because these are the molecules which will activate certain other specific proteins required in that particular pathway. Depending on the structure or chemical structure of the second messenger can either diffuse through cytosol or it can stay embedded in the lipid bilayer of the membrane. So, the secondary messenger, which is produced by binding of a ligand or a signaling molecule to the receptor, it can either diffuse away or it can stay there in position to activate the downward set of enzymes. Now, this particular panel shows what exactly happens, what are the two mechanisms by which the signal can be transferred from outside to inside. The left panel shows the binding of the molecule, which is blue circle, to the transmembrane receptor, and the cytoplasmic tail of the transmembrane receptor is bound to the effector. Effector molecules get activated and it leads to production of a secondary messenger, which then activates a series of protein and ultimately it leads to activation of the target protein. Second mechanism is the ligand binds to the receptor and to the cytoplasmic tails are attracted certain other proteins, but overall the next step is activation of other set of proteins which finally lead to activation of the target protein. So, these are the two mechanisms through which the signal can be transferred, and these mechanisms in total lead to various other processes like transcription, protein synthesis, cell death also. So, these are the mechanisms through which these processes will occur in the cell. Now, what exactly is happening here? When the signal is getting transferred, there is a change in the conformation of the protein. Proteins in the signaling pathway will alter the conformation of the protein downwards, which are in the cascade in the downward direction. So, this alteration can be done by modification of protein, and this is usually done by proteins known as protein kinases and phosphatase. Now, as the name suggests, protein kinases are those proteins which attach phosphate group to the protein. Attaching of this phosphate group by the protein kinases to the target protein leads to a change in the conformation of that particular protein and in most cases their activation. And these proteins will be deactivated later on by enzymes phosphatases, which remove this particular phosphate group which has been added to the protein. Human genome encode about 500 different protein kinases and approximately 150 different protein phosphatases. So, their importance can be gauged from this particular fact that the genome has such a large number of protein kinases and phosphatases which are required for conformation change of the protein and thus effective signal transduction from outside to inside. But most of these protein kinases are able to transfer phosphate group to either serine or threonine residues, which are two types of amino acid. But there are certain kinases which can also phosphorylate tyrosine residues in the protein. And some protein kinases they might occur as soluble form or other might occur as integral membrane protein. So, their position can change. It's not fixed that the kinases and phosphatases have to be cytoplasmic soluble protein. They might be transmembrane protein also, and these kinases might phosphorylate a large number of amino acids, different types of amino acids. Now, this phosphorylation has the ability to either activate or deactivate the enzyme, and the reverse process will act in a reverse manner. Suppose phosphorylation activates an enzyme, then dephosphorylation will deactivate that particular enzyme, and vice versa. Phosphorylation can also lead to an increase in protein-protein interaction. If a protein is phosphorylated, it might form contact with some other protein. It can also induce a protein to move from one subcellular compartment to another. There are a number of organelles within the cell. So, phosphorylation might cause protein to move from one compartment to another, and this event, that is phosphorylation, can also cause protein to undergo the degradation pathway. That is, proteins can be degraded if they are phosphorylated. So, there are a number of things which can happen if a protein is phosphorylated. Now, we know that I've already told that signaling pathways consist of a series of proteins which are activated one after another. Each protein in a pathway is able to alter the conformation of the next protein, and that's why it might get activated. Now, this conformation, as I've already told, is usually altered by phosphorylation. The target proteins ultimately receive a message which started from the ligand binding to the receptor, and target protein brings about the changes which are required by that particular cell, and which information was being transferred by the signal molecule to the cell. This entire series of events is termed as signal transduction. The information being carried by the ligand is ultimately passed on to the target cell, and it carries out processes required thereof. So, this particular panel shows how there are a series of proteins or enzymes which get activated one after another by the action of enzymes which are situated in the same cascade. So, protein kinase 1 activates protein kinase 2 by phosphorylation, which activates protein kinase 3, which activates a transcription factor which goes and binds to the DNA and starts transcription of the gene, leading to formation of mRNA. This mRNA will then be translated into protein, and the required and whatever that proteins, whatever is the function of protein, that will be caused by that particular protein. So, starting from the receptor from the signal binding, we have the end product, which is formation of a specific protein required by that particular cell. Now, there are a large number of extracellular messengers which are present in the body, and which can function to transfer information. These messengers include small molecules as well as large molecules. Now, these small molecules like amino acids and their derivatives like glutamate and glycine, they act as neurotransmitters as well as hormones. And there are other small molecules like nitrogen oxide and carbon monoxide, which are gaseous molecules, which can also function as signaling molecules. Now, going into detail with that, these neurotransmitters, they are able to carry signals between neurons and from neurons to other target cells. So, neurotransmitters, basically we know they are released at the end of the neurons and axon of the neuron, and they are able to transfer information from one neuron to another as well as from one neuron to other cells. A number of such neurotransmitters exist like acetylcholine, dopamine, epinephrine, etc., which are able to transfer information from neurons to cells or from neurons to other neurons. The arrival of action potential leads to release of neurotransmitter. We know how exactly this happens. We know how exactly neurotransmitters are released. Neurotransmitters are released from the ends of the neuron only when action potential reaches the ends. And once the neurotransmitters are released, they bind to the receptor on the surface of the target cell. Binding of neurotransmitter might activate a number of ligand gated ion channels. That means as soon as neurotransmitter bind to the receptor on the target cell, number of ion channels might open, which will lead to depolarization and creation of action potential in the target cell. Whereas some receptors which bind to the neurotransmitter, they might not lead to opening of an ion channel. They might lead to activation of G proteins, which are coupled with these particular receptors, and this can later regulate ion channel activity. There are hormones like peptide hormones, which are not very big in size, and growth factors. As we know that peptides are the widest variety of signaling molecule present in the body, ranging in size from a few amino acids to more than 100 amino acids. Peptide hormones like insulins, neuropeptides like growth hormones, and a diverse array of polypeptide growth factors can act as signaling molecules. And these neuropeptides, they are usually secreted by neurons, some example of which are enkephalins and endorphins. Now, there are polypeptide growth factors which control animal cell growth and differentiation. There are PDGF, platelet derived growth factor, which are required in wound healing. There are cytokines which are required for regulating development and differentiation of blood cells and controlling the activity of lymphocytes. There are membrane anchored growth factors which remain associated with the plasma membrane, and these peptide hormones and neuropeptides and growth factor, they all bind to cell surface receptor. They have to have receptor on the surface for binding them. Nitric oxide is another molecule which is a paracrine signaling molecule because its half-life is very less and it cannot travel long distances. It is released in the nervous system, immune system and circulatory system, and it is formed by nitric oxide synthase utilizing the amino acid arginine. This particular gas is able to diffuse through the plasma membrane of the target cell, and it binds to the heme group at the active site of guanylyl cyclase, and the end result of this particular, or the secondary messenger of this particular signaling molecule is production of cyclic GMP. This signaling molecule may also cause nitrosylation of cysteine residues in the target protein. Release of neurotransmitter from the end of the nerve cells causes synthesis of NO or nitric oxide by the endothelial cells, which diffuses to the smooth muscle cells and activates guanylyl cyclase producing cyclic GMP, which leads to smooth muscle relaxation and blood vessel dilation. Another gas which is used as a signaling molecule is carbon monoxide, which functions as a signaling molecule in nervous system. CO is produced in the brain cell and it also stimulates guanylyl cyclase, which again produce cyclic GMP. Other extracellular messengers present in the body are steroids, which are derived from cholesterol, and eicosanoids, which are derived from arachidonic acid, which is a lipid. Now, steroid hormones basically they are able to cross the membrane and they bind to molecules or the receptors which are present in the cytoplasm, and when once they are bound to these receptors, which are basically transcription factor, they can go and enter the nucleus and activate specific set of genes. Thyroid hormones, which are not similar to steroid hormones in structure, vitamin D3, which is again very different in structure, and retinoic acid, which is again a different type of molecule, they all function in similar manner. That is, they can easily traverse the plasma membrane of the cell and bind the receptor molecules present in the cytoplasm. All these steroid hormones are derived from the building block, which is the cholesterol. Cholesterol is the initial molecule from which other steroid hormones like testosterone and estrogens are derived. Now, these molecules diffuse across the plasma membrane, bind to the receptors, and then these receptors, which are transcription factors, they go and bind to specific elements in the hormone response elements in the DNA and activate specific set of hormone responsive genes. Glucocorticoid receptor, which is bound to HSP90 chaperones in the absence of hormone. Once the hormone binds to these particular receptor, it dimerizes and leads to activation, transcription of the target gene. Again, thyroid hormone is another type of hormone which is bound to a co-repressor which represses the transcription of gene. But binding of hormone leads to removal of this co-repressor, and this leads to interaction of the hormone with co-activator. And this again induces transcription of a specific set of genes which are related to this particular hormone. Then the other class of molecules which can be used as signaling molecules are eicosanoids. These eicosanoids are basically synthesized from arachidonic acid, which again is derived from phospholipid. Now, these eicosanoids include a large variety of molecules like prostaglandins, thromboxanes, and leukotrienes. All of them either function in autocrine or paracrine signaling pathway. They do not function in endocrine pathway. Either autocrine, that is, they activate or they regulate themselves, and paracrine, that is, they regulate the cells or they send signal to cells which are in their vicinity. These molecules, they can stimulate blood platelet aggregation, they can regulate pain, they can regulate pain sensation, they can regulate inflammation, blood pressure, blood clotting, and smooth muscle contraction. Arachidonic acid is basically converted to prostaglandin H2 by enzyme cyclooxygenase, and this enzyme cyclooxygenase is the site of action of a number of drugs like aspirin and other non-steroidal anti-inflammatory drugs, which inhibit the production of prostaglandin, which are responsible for pain sensation. So, this is how certain drugs act, and they help us in numbing the pain sensation. Now, apart from these signaling molecules, there are various other polypeptides and protein which can act as extracellular messengers. Some of them are transmembrane proteins, while they can be found associated with elements of the extracellular matrix. A large number of proteins get excreted into the extracellular environment, where they are able to regulate processes such as cell division, differentiation, immune response, cell death, and cell survival. So these are the various types of messengers present in the body. Now these messengers need to have certain specific receptors for binding them and transferring information. The various types of receptors are G protein coupled receptors, receptor protein tyrosine kinases, ligand gated channels, steroid hormone receptors, specific receptors such as B cell and T cell receptors. Now, G protein coupled receptors are the family of receptors which contain a 7 transmembrane alpha helix group, and they translate the binding of extracellular signaling molecule into activation of GTP binding protein, that is G protein. We have receptor protein tyrosine kinase which bind the ligand, and this leads to dimerization of receptor, which is followed by activation of the receptor's protein kinase domain, which is present in the cytoplasmic region. Upon activation, these protein kinase phosphorylate tyrosine residues of the cytoplasmic substrate and then activate a series of proteins. Ligand gated channels basically they conduct the flow of ion across the membrane, and once the signal is bound to the receptor, they can open specific ion channels which can lead to change in the voltage across the membrane. This sequence of events is the basis of formation of nerve impulse. And then we have a large number of receptors which can function by specific or unique mechanism. Some of these receptors, like B and T cell receptors, are involved in response to foreign antigen, and they associate with the known signaling molecules such as cytoplasmic protein tyrosine kinases. So these receptors basically act by binding to the specific ligand, and they initiate a cascade of reaction which ultimately leads to target protein activation, which could be a transcription factor or it could be a protein in itself, and this protein will carry out the function required for that particular cell. Thank you.
With this note, thank you, sir. Thank you so very much for giving us this session. Friends, you are requested to be with us as we are back after a short break and would be discussing more. Thank you. Hello friends, welcome back to this session. Friends, as you know that in our previous session we discussed on cell signaling, whereas in this half we are going to talk on G protein coupled receptors, and for the discussion on the topic we have with us in our studios Dr. Varunendra Singh Rawat. Dr. Varunendra Singh Rawat is Assistant Professor in Department of Zoology, Hindu College, University of Delhi. So let's welcome our guest Dr. Rawat and let's try to understand more on the topic. Hello sir, welcome to the lecture.
Thank you. So, I'll be talking about certain receptors which are termed as G protein coupled receptors in this particular lecture. As we know, cells communicate with each other through release of signal molecules these signal molecules are intercepted by specific target cells, and these target cells are then they undergo a series of reactions which bring about desired change in these target cells. It is essential for cells to survive that they be able to communicate with their environment, be it the extracellular environment or be it the cells which are present in their vicinity, or even if they are not present in vicinity, they are present far off. For example, hormones which are released by specific organs like pituitary, they have to communicate with organs which are far off. So they release signal, that is hormones, and these hormones need to be intercepted by the target cell. Cell signaling is imperative or essential for survival of the cells, survival of the tissues, and survival of the complete organism. And this particular signaling and receptor of the signals is dependent on a certain set of proteins. These proteins are termed as receptors. In this particular topic, I will be talking about receptors which are termed as G protein coupled receptors. Why they are called G protein coupled receptors, I will be discussing that. And I will be also discussing specific proteins known as G proteins which are attached to these G protein coupled receptors, and the end product which they produce, which is cyclic AMP, and how this molecule is responsible for carrying out a number of functions in the body. So by the end of this lecture, we should know what exactly is a G protein coupled receptor, we should know how the G proteins function, and we should know about the role of cyclic AMP in the different types of cells. Now, the receptors could either be present on the membrane of the cell and they can bind the ligand and they can initiate a set of reactions afterward, or receptor might be found in the cytoplasm, like receptor for the steroids, which can bind the steroid molecules which easily pass through the membrane of the cell. But most of the ligands, they have their receptor present on the surface of the cell. And this leads to a series of changes which could either be opening of the ion channels, which could be change in the conformation of the protein itself, receptor itself after binding of the ligand or the signal, and this conformation might lead to changes in other set of proteins. Ultimately, the target protein will get activated or deactivated. Now, what exactly are G protein coupled receptors? G protein coupled receptors go by the acronym of GPCRs, and these are so named because these receptors interact with G proteins in their cytoplasmic domain. These GPCRs are also referred as transmembrane, 7 transmembrane receptor, because there are 7 helices which pass through the membrane of the cells for formation of this particular receptor. Basically, this is a protein, and 7 helical regions cross the membrane of the cell wherein this particular receptor is present. This particular family of receptor is a very large family. That means a large number of genes are present in the genome which code for a large number of GPCRs, and this is the single largest superfamily of proteins being encoded by animal genome. So their importance can be gauged from the fact that this particular family of proteins is the largest family of proteins being encoded by the animal genome. Thousands of different G protein coupled receptors have been identified in organisms which range from the unicellular yeast to plants and to mammals. So from unicellular organism eukaryotes to multicellular organism, G protein coupled receptors are found in all of them, and the number varies from 100 to 1000. The genome of organism C. elegans has been sequenced, and it has been found that this particular organism's genome has about 1000 genes which code for different type of GPCR. And these different GPCRs regulate a different set of array of the cellular processes. Now the question arises, these GPCRs, as the name suggests, they are bound to G proteins, then how are they able to cause an setting up or starting of a different set of cellular processes? How exactly that happens? Before going into that, we should know that G protein coupled receptor can bind a large number of ligands, that is, a number of natural ligands are able to bind to these receptors, and these signal molecules which can bind to receptors range from hormones to neurotransmitter, to opium derivatives, to tastins, to chemoattractants and odorant. So there are a large number of signal molecules which can bind to GPCRs, which again are being coded by a large number of genes. So that it means that there is a variety in GPCRs and a variety in the signal molecule which they bind. This particular panel shows us a set of ligands which are bound by GPCRs. For example, epinephrine binds to beta adrenergic receptor, which is a type of GPCR which activates adenylyl cyclase, and the physiological response for this particular activity is breakdown of glycose. Similarly, we can see acetylcholine, which again binds GPCR, which is muscarinic receptor, goes by the name of muscarinic receptor, which again activates potassium channels, and the physiological response of this particular binding is slowing of pacemaker activity of heart. So this table tells us that there are a plethora of GPCRs which are present in cells, and they can bind a number of ligands or number of signal molecules, and they can bring about a number of physiological changes in the cells. What exactly is the location of GPCR? As I have already told, they are present in the membrane, and there are 7 helical regions which span the membrane. The amino terminals of G protein coupled receptor is pointing toward the outside of the cell, that is toward the extracellular region, and the carboxyl terminal points towards the inside of the cell, that is toward the cytoplasmic site. There are 3 loops present on the outside of the cell of these alpha helical region. 3 loops are present on the outside, and these 3 loops together form a ligand binding pocket, wherein goes and bind the specific ligand or signal which will activate the corresponding GPCR. Also on the inside are present 3 loops, and these are the 3 loops which will bind the intracellular signaling protein. So we have 3 loops toward the outside which will bind the ligand molecule, and 3 loops toward the inside which will bind the intracellular signaling molecules, which will start a cascade of reactions. G proteins, as the name suggests, G protein coupled receptors, because they bind G proteins. G proteins are able to bind to the 3rd intracellular loop toward the cytoplasmic site. And there are a number of other proteins which can bind to the carboxyl terminal of these GPCRs, and these proteins then can link the receptor to the other signaling molecules and the other effector molecules in the cell. So the cytoplasmic tail or the cytoplasmic domains are very important, because they are able to bind the effector molecules, and they are also able to bind other proteins which can activate the other effector molecules. So there is direct binding of effector molecules, or there is binding of other proteins which activate certain other effector molecules in the chain or the cascade. These GPCRs, they can occur either in active conformation or they can occur in inactive conformation. The inactive conformation, that is the state where GPCR is not bound to the ligand molecule, it is stabilized by interaction, non-covalent interaction between amino acid residues in the transmembrane alpha helices. When the ligand binds to these receptors, it disrupts these interaction between the alpha helical regions, and because of this particular disruption, there is change in the conformation of the receptor, and the conformation changes from inactive to active conformation. This particular change in conformation results because of rotation or shift of the transmembrane alpha helical region. As I have said that the inactive conformation is stabilized by non-covalent interaction between these alpha helical regions. Once the signal molecule binds to the receptor, there is rotation of the alpha helical regions, and these interaction break down, and the conformation of the receptor molecule changes from inactive to active. As the alpha helical regions rotate, because they are in continuity with the cytoplasmic domain, the alpha helical regions rotate, and they also cause rotation of the cytoplasmic loops, which in turn lead to an increase in the affinity of the receptor for G protein. which are present on the cytoplasmic side of the plasma membrane. So summing up, what exactly happens? A ligand or a signal molecule comes and binds the GPCR. When it binds to the GPCR, the conformation which was inactive, it changes into active conformation because there is no more interaction between the alpha helical region, and alpha helical regions rotate, and this leads to change in the conformation of the cytoplasmic loops, and the cytoplasmic loop conformation changes, which is able to recruit G proteins towards the cytoplasmic loops, and once G protein is recruited, it leads to activation of certain other proteins in the cascade. The ligand bound receptor formed a receptor G protein complex. The interaction with the receptor induces a conformational change in the alpha subunit of G protein. Now these G proteins are heterotrimeric, that is, there are 3 polypeptides present which are all different from each other. These are alpha, beta and gamma polypeptide. Coming together of alpha polypeptide, beta polypeptide and gamma polypeptide leads to formation of G protein. Once the receptor, G protein coupled receptor is bound to ligand, it is able to recruit the G protein. The alpha subunit of G protein, which in inactive state is itself bound to GDP, because of its recruitment by the G protein coupled receptor to the cytoplasmic side, the alpha subunit of the G protein, it exchanges its GDP with GTP. In the activated state, a single receptor present on the membrane can activate a large number of G protein molecules, and these G protein molecules in turn can lead to activation of a large number of effectors, and thus this leads to amplification of a signal. A single signal molecule getting bound to receptor can activate a large number of G protein molecules, which then can activate a large number of effector molecules. So basically an amplification of signaling is occurring in this particular process. Now this particular panel shows the arrangement of the G protein coupled receptor, which can be seen as pipes present in the transmembrane domain. They have a domain which projects towards the outside or the extracellular region, and domain which are present towards the cytoplasmic side. Now the ligand can come and bind to the cytoplasmic side, which leads to change in conformation of the cytoplasmic domain, leading to binding of G protein, wherein the alpha subunit of G protein exchanges its GDP with GTP. This G protein then can activate the effector molecules, which are usually present in the intermembrane region, in the membrane itself, and these effector molecules then can activate a set of protein, which leads to production of intracellular second messenger. These second messengers are the one which will then activate other proteins, and a series of such reactions will take place till the target protein is activated. This particular panel shows inactive and active form of rhodopsin. Inactive form occurs whenever the ligand is not bound, and active form occurs when the ligand is bound to the rhodopsin molecule. Summing up the events till now, we see in this particular panel the step 1 where receptor is not bound to ligand. After binding of the ligand, it recruits G protein, which has 3 subunits, alpha, gamma and beta. Alpha subunit is the one which can either bind GDP or GTP. This alpha subunit bound to GDP gets recruited by ligand bound receptor. Once it is bound to the ligand bound receptor, alpha subunit exchanges its GDP with GTP. Once that happens, alpha subunit separates from the 2 other subunits, namely beta and gamma subunit. The alpha subunit is still bound to GTP. Now it goes and it activates effector molecules, and this effector molecule is basically an enzyme in itself. It leads to production of secondary messenger. In this particular panel shows there is production of cyclic AMP by utilizing ATP molecule. Once this happens, after this happens, the GTP, which is bound to GDP, it hydrolyzes. GTP, which is bound to alpha subunit, it hydrolyzes, and it leads to formation of alpha subunit, which is bound to GDP. Now, this particular alpha GTP is it is having affinity for beta and gamma subunit. It goes and binds to them, and again it is available for binding to the receptor, which is bound to the ligand. Now, this particular signaling cannot go forever. It has to be stopped, because if if a ligand is bound to the receptor and signal keeps on producing secondary messenger, then it will lead to problematic situation. So there are other proteins which will lead to phosphorylation of the cytoplasmic tail of the receptor, which will end the transfer of signal from the outside towards the cytoplasmic tail of the receptor. Or there are other molecules known as arrestin, which can go and bind to the cytoplasmic site of the receptor and end the process of signal transduction. Now, what exactly, I have already told what exactly are G proteins. These were first characterized by Martin Rodbell in 1917. As already told, these can either bind to GDP or they can bind to GTP. They are heterotrimeric. They have 3 different polypeptides, and they differ from other such G proteins like RAS proteins, which are having only one polypeptide. These G proteins are held in place by lipid chains, held in place they are found attached to the plasma membrane, because they are bound by lipid chains to the alpha and gamma subunit. And only alpha subunit is the one which can bind GTP or GTP. Now, G alpha subunit undergoes conformational change whenever it interacts with the activated GPCR. Then it exchanges GTP with GTP and dissociates from G, beta and gamma subunit, and then activate adenylyl cyclase. This adenylyl cyclase leads to production of CAMP. Apart from production of CAMP, there are other effectors. That is, apart from adenylyl cyclase, there are other effectors which can get activated by G proteins, and these are phospholipases C beta and cyclic GMP phosphodiesterase. And the end result of activating of these effectors is that second messengers are produced, which initiate a set of reactions. Now, G alpha is the one which can bind GDP and GTP. Hydrolysis leads to change in conformation. Once GTP which is bound to G alpha, once it hydrolyzes, this G alpha will go and bind to G beta and gamma, and it will again start a series of binding to a receptor which is bound to ligand. Now, these G proteins, they can be classified according to the function they perform. Now, these G proteins have been classified into 4 types, namely GS, GQ, GI and G12/13. Now, what exactly is the basis of classification? This classification is based on the G alpha subunit and the effector to which they couple. That means all the type of G proteins, they have been classified into 4 types, and the basis of classification is what exactly to which effector are exactly G alpha subunit are binding after they are bound to the G protein coupled receptor. The response which is elicited by an activated GPCR, it always depends on the type of G protein with which it interacts. Suppose a ligand comes and binds to type A cells, and similar ligand goes and binds to type B cells. Both have G protein coupled receptor, but the effect which they show is different. Why that is different? Because the G protein coupled receptor, it will bind to a specific type or specific class of G protein. Now, that particular specific class will be able to activate specific type of effector molecules, and these effector molecules in turn will activate will produce specific secondary messenger. So, end result will be different in cell type A and cell type B, because G protein coupled receptors were recruiting different types of G proteins, and thus they were activating different types of effectors, and therefore they were producing different types of secondary messenger. Thereby they were activating different types of signaling pathways. Now, coming to types of G proteins, GS family member or GS family of the GS family protein, they couple only to adenylyl cyclase, whereas GQ family members, they contain G alpha subunits which can activate PLCB. Now, what exactly PLCB does? It hydrolyzes phosphatidyl inositol bisphosphate. Hydrolysis of phosphatidyl inositol bisphosphate leads to production of inositol triose phosphate and diacyl glycerol, which are secondary messenger quite different from CAMP, and these secondary messenger produce a different set of reaction as compared to cyclic AMP. Then we have GI subclass. Now, these activated GI subunit, they function by inhibiting adenylyl cyclase. So, they differ from GS subunit because they can inactivate adenylyl cyclase, whereas GS type of G proteins, they activate adenylyl cyclase. Now, the last type is G12-13 type. These members are less well characterized as compared to the other classes, other family, but it has been seen that their inappropriate activation leads to excessive proliferation of cells and can lead to malignant transformation, that is formation of cancerous cell. After the G alpha subunit has dissociated from the beta gamma complex, it has been found that the beta gamma subunits of the G protein, they can also take part in signaling function, and they can couple to a number of different types of effectors like PLCB, K1 and Ca2 plus ion channels, and also adenylyl cyclase. So, all the three subunits can take part in signal transduction. After there has been activation or the transfer of signal, these receptors have to be desensitized. That means transferring of signal has to stop at some point of time. This is termed as desensitization. Now, this desensitization, this can occur by blocking the active receptor from turning on more additional G protein. This can occur by activating G protein coupled receptor kinases or GRKs, which phosphorylate the cytoplasmic domain of GPCR. That means no more G protein will be activated by these receptors. Or proteins termed arrestin, they can go and bind to the cytoplasmic tail, and herein they compete with G protein. So, the site is occupied by arrestin, no more G protein can come and bind to this particular site. Arrestin molecules are also capable of binding to AP2 adapter molecules or AP2 proteins, which are situated in the clathrin coated pits near the membrane of the cell. Interaction between arrestin and the clathrin coated pits leads to uptake of the phosphorylated GPCR into the cell by endocytosis. So, arrestin can lead to endocytosis of the GPCR which are phosphorylated, and these receptors which have been removed from the surface, they have several alternative outcomes. What are those outcomes? In some cases, these receptors travel along the endocytic pathway into endosomes, where the arrestin molecules serve as a scaffold for the assembly of various cytoplasmic signaling pathways like MAPK pathways. So, this is one course of action. In the second outcome, the internalized receptor, they may take these receptors from the endosome to lysosome for degradation. And the third outcome is these arrestin bound GPCRs, they may be dephosphorylated, because the phosphorylated receptors are inactive. These GPCRs may be dephosphorylated and they may be taken back to the plasma membrane. So, these are the various outcomes of the GPCRs which are bound to arrestin and which are endocytosed by the cell. So, this particular panel shows what are the three different options. They can either be taken by the lysosome, they may be recycled, or they may take part in certain other activities like activating a series of enzymes. Now, the G alpha subunit which is bound to GTP, it is active only when it is bound to GTP. It can become inactive once GTP is hydrolyzed to GTP. So, this GTPase activity is itself present in the alpha subunit. The strength and duration of the signal which is being transmitted depends on the rate of GTP hydrolysis by the G alpha subunit itself, and this particular response is accelerated. Termination of response of the activity of G alpha subunit is activated by certain proteins which are termed as regulators of G protein signaling or RGS. Summing it up, the mechanism of transmitting signal across the plasma membrane by G proteins is very necessary, and it's necessary. It can be seen because it is found in all the organism, and it is of ancient evolutionary origin, and it is highly conserved. So because it is of ancient origin, it has been utilized by certain bacteria for gaining entry into the cell, by certain bacterial toxins for gaining entry into the cell, like cholera toxin and pertussis virulence factors. These are the toxins which target GPCRs and G protein. G alpha subunits are modified once these toxins are bound. Their GTPase activity is inhibited, and once the cholera toxin is bound to the GPCR, and this leads to the activity of adenylyl cyclase producing large amounts of cAMP, leading to production of large volumes of fluid which are released into intestinal lumen, which might also cause dehydration and death of the individual. Pertussis toxin, in contrast, it inactivates G alpha subunit. The signaling pathway leading the host to mount a defensive response against the bacterial infection is inhibited whenever pertussis toxin is bound to the GPCR. So this is how bacteria manipulates the system of the eukaryote in infecting them and producing disastrous results. G protein coupled receptor has also fetched a Nobel Prize to two scientists going by the name of Linda Buck and Richard Exel. In the year 1991, Linda Buck and Richard Exel, they were able to identify a large family of G protein coupled receptors which were responsible for detection of odors and recognition of these different types of odors. They were awarded Nobel Prize in Physiology in 2004 for their work. So G protein coupled receptors are also responsible for deciphering different types of odors which we are able to sense from our environment. There are number of disorders which are related to G protein coupled receptor if there is any mutation or malfunctioning of the G protein coupled receptors. For example, retinitis pigmentosa. It occurs whenever synthesis of rhodopsin molecule is faulty, and because of faulty rhodopsin molecule, the G proteins cannot be activated, and because G protein cannot be activated, the colors cannot be deciphered by the individuals, the retina of the individual. Second disorder is thyroid adenoma. Now, thyroid adenoma is basically, it occurs when TSH receptors have an altered third intracellular loop, which leads to continuous activation of G protein. Now, because of continuous activation of G protein, there is an excessive thyroid hormone secretion, and because of thyroid hormone secretion, it leads to activation, increase in metabolism, and activation of various types of cells, and it might also lead to cancers of different types. So we see here that G protein coupled receptor is very much imperative for normal functioning of the cell, but if it is functioning, if the functioning goes awry, or if there is malfunctioning, it can lead to production of a large number of diseases in the multicellular organism. Thank you.
So thank you so very much for giving us this session. Friends, you are requested to write to us at info.cec@nicc.in if you have any queries or feedback. We will be meeting again soon and would be discussing more in the series. Till then, take care. Goodbye. Thank you, sir. Thank you.